Hi there. So I'm describing a image analysis exercise I developed for a microbial physiology lab. It's a 4,000 level class consisting of upper level undergraduates, primarily majoring in biology and some in subdisciplines like microbiology. Anyway, one of the tools that I used for the image analysis is listed here, the GIMP software. There's the URL for downloading it. It's shareware, it's free, it's pretty straightforward to use, and it also has most of the features of Adobe Photoshop if you're familiar with that. So it's really versatile. Okay, so the example I'm showing here is from a microbial physiology class last spring when it transitioned in a panic to online format. I wanted to have some sort of experiment that the students could do from the safety of their homes using their computers. So dovetailing with other things that we'd studied over the semester in the lab, I decided to have them do an image analysis project of transmission electron micrographs where they quantified the abundance of these subcellular structures called carboxysomes in bacterial cells. So what I did was I collected a whole bunch of transmission electron micrographs for this project. Now the image analysis project that I'm describing here was done on electron micrographs, but it could really be done at any sort of collection of any sort of image. So the big question we were addressing with our project was, does carboxysome abundance in cells change with different growth conditions? So we had a series of electron micrographs of cells grown under a variety of conditions, and the question was to quantify the number of carboxysomes, which are visible in this electron micrograph as round inclusions, whether the number of carboxysomes changed in cells under different growth conditions. So I began with the electron micrographs. I had the students all in a Zoom meeting, in a Teams meeting with me, and I screen shared with them, and I showed them a series of electron micrographs to kind of educate them on what to look for. So the first slide here shows a variety of cells that have been numbered. So I walked through the different examples with the students. I said, okay, here's this first cell, number 1. Do you see any carboxysomes there? Why do you think it's all dark? Turns out this one, the slice of the cell is an edge slice, so you don't see any cytoplasm there. Cell number 2, you do have a transverse section of the cell, and you do see some inclusions that could be considered carboxysomes. 3 is another glancing blow like 1 is. 4 is a good transverse slice. 5 is the one where you have the most obvious carboxysomes. So I walked through some examples with the students and then showed them another micrograph and had individual students over Zoom or over Microsoft Teams tell me which cells had carboxysomes and how many. From there, to make sure each individual student was clear on what to look for, I provided them with an image on Canvas and also provided them with a spreadsheet, and individually they had to fill out the worksheet to tell me which cells had carboxysomes. Once I'd taken a look at all of their worksheets, I could feel pretty good that they knew what to look for in these images. I also told them how to get the image analysis software from the web and download it onto the computers. I also provided them with an image on Canvas that could only be opened by GIMP, and they wouldn't know what the image depicted until they opened it with GIMP. So I had them download the software, I directed them to an image to open, and then I had each of them individually email me to tell me what the image was a picture of for credit, to make sure that the software was downloaded correctly and that they could actually open images with it. So the next week, I actually walked them through the process of image analysis. Now that they knew what to look for in the images, and now that I knew that the software was correctly installed on their computers, it was time to show them how to do the actual measurements. So each student was assigned 4 or 5 images. The images were made available on Canvas, and also a workbook for entering the data by each individual student, and also to make calculations was available on Canvas. The students are guided on how to make the measurements. Using GIMP, I went through I opened up an image with GIMP and I showed them how to use a drop-down tool called Measure that allowed you to actually measure things in a window in GIMP. Now you notice that each image that I have has a yardstick in it, and that allowed the students to calibrate how long a line they would draw in their image was. It could convert pixels, which are displayed in the measure feature of GIMP down here, into microns by basically figuring out how many pixels the yardstick was and then using the appropriate math to convert the pixels into microns. So I guided them through the process of calibrating the image with these first 3 slides, and then I went through the process of applying what they'd learned to actually do these measurements to a cell. I showed them how to measure. Well, number one, I had them start off with counting the carboxysomes in each individual cell, which they already knew from their previous week's exercise. And then together we measured the length and width of a particular cell that we counted the carboxysomes in and agreed on a length in pixels and a width in pixels. And then I showed them how to enter those values into their spreadsheet. So I gave them step-by-step instructions on how to use GIMP and how to use GIMP to measure things in an image. Then what I had them do is I had them guide me through analyzing a few cells. I said, okay guys, now we're going to pick a cell. You're going to tell me, how do I start? And they said, okay, first you're going to count the number of carboxysomes. And I would say, great, how many carboxysomes? And they would say, oh, about 5. Great. Well, how do I enter those data? Well, you enter up, so it was step by step. I had them tell me what to do. And that really helped figure out where points of confusion might be. So I had them analyze a few cells with me in real time. And then I kept the Microsoft Teams meeting open, and I had the students continue on their own computers for about 20 minutes, and I was there to answer questions. A few questions did come up. Sometimes they wanted me to verify that they were measuring correctly or counting correctly, and that was fine. After about 20 minutes, it seemed like everybody'd settled in. After that, the students were on their own and responsible for uploading their workbooks. And then I combined all of their data into a single data sheet. The following week was a wrap-up. I took all of the data from the many students and took a quick look at it to make sure that things had gone according to plan. And I coalesced it into a single large data set. The next time we met, we talked about how we could graph the data to tell a story, just basic how to do science stuff. We summarized the data. We talked about what was there and what it meant. And then, of course, we suggested follow-up experiments to carry things forward given the opportunity to do so. So the key bits, I started off with the key bits for doing an image analysis project with the students virtually. I think the key steps here is to start off by helping the students understand what to look for and also what to avoid. These are really great places to discuss the limitations of the techniques and what the artifacts might be in the image. Another key step is to practice an image as a group in class for the shy people. The group work is good because they can kind of see the pitfalls. They might be too self-conscious to put themselves out there, but they can understand, how this works a little better by hearing more extroverted or daring students be making the mistakes and learning how to rectify. And then to make sure the individuals are getting it, the practice image quiz is good. I kept this low stakes by keeping the points value low enough that it wouldn't slaughter somebody if they totally didn't get it, but high enough to make them be sure to give it a good try. I then gave them a very step-by-step introduction to the spreadsheet and the math, and a step-by-step introduction on how to use the software. Then I collated the data myself, but then we spent the time to come back and discuss the assembled data set together. Okay, so that's my description of the image analysis project that I designed in a panic for microbial physiology, but it is something I intend to continue even if I don't teach that class again remotely, and hopefully there's all kinds of projects that one could do with image analysis with the students. The great thing about GIMP is that it's pretty easy. I didn't have any problems with students being unable to use it to do the measurements I described, and it's also free. So all good traits. Anyways, I hope this was helpful.